Let me talk to you very briefly about Rusty Gage. I could spend a long time introducing him, but I'll try to do it quickly so he can get on the podium here. In 2003, Time magazine profiled 100 individuals who most influenced the last 100 years. Among those listed were noted scientists, including people that you've heard of, like Einstein and Freud and Salk, and today's speaker, Fred, known affectionately as Rusty Gage. Dr. Gage is a professor in the Laboratory of Genetics here at the Salk Institute. He received his bachelor's degree from the University of Florida and earned his PhD at Johns Hopkins University. He has received numerous awards, such as the Charles A. Dana Award for Pioneering Achievements in Health and Education, the Christopher Reeve Research Medal, the Max Planck Research Prize. He's also a past president of the Society for Neuroscience and was elected to the National Academy of Sciences in 2003. Until quite recently, neuroscientists were quite convinced that the neurons, or brain cells, we are born with are all the brain cells we would have during our lives. Then, in 2001, Dr. Gage and his laboratory showed in a groundwork-breaking set of experiments that neurons are constantly being born, particularly in the learning and memory centers of the brain. His discovery forced scientists to rethink some of their most basic ideas about how the brain works. Today, neurobiologists no longer argue about whether or not the brain can grow new cells. Instead, they are trying to figure out how these cells, this cell growth, can be harnessed to treat everything from epilepsy to stress to depression. One area that holds enormous promise is stem cell replacement of nerve cells that die prematurely in our brains, mostly due to pathological conditions or disease. The majority of the citizens of this state believe in this promise and have voted to support efforts to make this promise a reality. To move in this direction requires that we understand much more about what stem cells are and what they do. Tonight, Dr. Gage will talk about some key aspects of this fascinating field of research in his lecture, which is entitled Stem Cells, the Brain's Beginnings.
Thank you, Eduardo. It's particularly a pleasure to be the first speaker of this Brain Gray Matter series, highlighting the neuroscience efforts here on the Mesa. One of the things that's always been very rewarding for me living here in San Diego and working, I must say, both at UCSD and the Salk at various times in my career has been the cooperation that exists of the scientists on the Mesa, and I think that's been one of the reasons why we've had the success that we have had here, is because of the ability to suppress the egos and get down to the basics of science. So that's been a continuing important effort, and I think this Gray Matter series will help us to reach out to the public and provide some of the knowledge that we've been gaining for their own edification. And it becomes even more important as we face this complex world that we live in now, where the public and private sector are in positions to make important decisions about what we will do and how we will use our resources in the future. And obviously I'm speaking very specifically now about the proposition issues here in California and the passing of Proposition 71, which some of you may be aware of. So I thought I would take this opportunity of the timing of this lecture to really split my lecture up into two parts. One is to discuss with you the general aspects of stem cell biology, but I really want to go into embryonic stem cells, human embryonic stem cells. What are they? What is the promise that's out there? What are the realities of what it is that the science will be able to deliver, and what will that look like? I will also pointedly take advantage of this audience to talk about some of my own work and my interest in adult stem cells. And this will, by contrasting the embryonic with the adult, we can begin to see the full range of things that are out there to be had. So just to summarize, I'll start off by getting down to some terminology of what really is a stem cell and what are different varieties of stem cells. A little bit about the basic biology of how these cells change and their fates and their potential functions. And I will spend a fair amount of time discussing the utility we have made here in the state of California to progress in our research towards some utility, and hopefully get some idea of this distinction between the adult and the developing brain. So I'm going to start off by talking about terminology, and there's really two components of a definition of a stem cell. A stem cell is a single cell that can self-renew. That means that when it divides, one of its progeny can give rise to itself. The other, we say daughter cell of that dividing cell, gives rise to and differentiate into a functioning cell. That's it. A stem cell has two properties. It can divide, give rise to itself, self-renewal, and it can give rise to some other functional cell. Well, if that were all it were, one wouldn't see where the controversy lies in dealing with stem cells. However, the problem has more to do with the words that come before it, and that is words that I'll go over with you. There's that describe the potential of the cells, and they also describe their derivation. So the most potent of all cells is called a totipotent cell, and that's a fertilized egg. Because a fertilized egg, when sperm and egg join through meiosis, you get a zygote that can give rise to all organs of the body, not just all cells of the body, but organs of the body, and a full organ, and a full functioning human being. Now, those of you who are listening very carefully will also note that this zygote really doesn't fulfill the first criteria of a stem cell. It's not self-renewing. It basically can give rise to several additional cells, which then are self-renewing. Nevertheless, you'll hear people call it the totipotent stem cell, but in fact, our terminology breaks down already. Most of the emphasis has been on embryonic stem cells, and embryo is different from a zygote by virtue of the fact that the cells of the zygote have divided multiple times and formed the initial stages of tissue called a blastocyst. And I'm going to go into more detail of what this is and what this looks like in a bit. But the difference between a totipotent cell and a pluripotent cell is that these cells of the blastocyst are these are the embryonic stem cells. These cells can be grown in culture, and they can give rise to all the cellular constituents of the body. So every cell of the body except for one particular cell type, which is called a placenta. So as far as we know, the embryo cells can't give rise to the placenta, but they can give rise to individual cells. The other thing that is missing in this, as we'll see, is that the embryonic stem cells we as scientists, and it's very difficult for them to give rise to any kind of structural organ, that they're great at giving rise to individual cells which have phenotypes or functions, but not to give rise to organs very easily. So they are, by virtue of that, more limited than the totipotent cell, which can give rise to everything. From a pluripotent cell, we move to a multipotent cell. And this distinction is important. This is after what we call organogenesis has begun. That's when the embryo now is moved into a fetal stage and organs are beginning to form, and the embryonic stem cells now differentiate quite importantly into cells of different organs.